important, and as good as the role is like zinc, where it involves all the 6 major metabolic processes, and it is probably it is available in the nature very abundantly, and it is the second most abundant element after that oxygen, and comparatively it is 28% of carbon is available. This availability is being constituted. But problem we have in the silica probably, because silica based most of our minerals present in the soils are silica based minerals, and most of these minerals are contributing silica, but they are not in the monomeric form. They are in the complex form. That's why probably the availability may not be suitable for the plant nutrition. So in such a cases, what happened? Whatever silica present in the earth crust is in geochemically is an inert material, and it may not be soluble for the plant system. Whereas the liquid phase of the system, probably we know it is there in the solid solution. It is a more complex, but we take it as an abundantly important element. And at the same time, what happened? The silica present in the most of the in a solution form, probably it ranges from almost 15 to 17 milligauss. And other most important aspects is it is being now considered as the most important beneficial element, and it is being designated as a alternative to the best nutrition for the global yield, and also plays a significant role with regard to the biotic and abiotic stresses, and also maintaining the balance in the nutrition aspects, and also plays a significant deterrent effect on the pests and diseases, and plays a significant role in most of the physiological processes like upregulation and downregulation. This is where it happens. It plays a significant role, especially biotic stresses like biotic factors like Insect-Pests. This is one area where really a lot of people they started working in this area. Probably in Japan and China, but probably India it is just a start probably in this regard. And we know that silica, because of this, probably in 2021. Based on their importance and requirement for silica fertilizations, our Indian government has approved silica as a beneficial organic nature, because we practice the silica as an organic, as it what you call biofertilizers. Because most of the export-oriented vegetable crops, let's say especially the zucchini, the crop which is highly detrimental for several insect pest diseases, especially in some of the diseases where silica plays a significant area. Because any export vegetable produced like zucchini, it is almost 100% export-oriented vegetable where it is being dissolved in vinegar and it is been exported, where it is being tested very rigorously spontaneous, but silica plays a significant role where it has residues with regard to the control measures with pest and disease. And you will see this chart, silica application. There are different sources of silica in the nature. I think now it is being industrial based products we have, and in nature also we have developed, because silica products developed from Australia, it is a ore based product and it is recommended for soil application. Besides, we have chemical based formulation like calcium silicate. This is also recommended for soil application in order to counteract the abiotic and biotic stress as well as, but in case biotic stress with other advantages are there. There are some of the sources where we use for silica nutrition or potassium silicate as well as orthosilicic acid. They are the industrial byproducts where we can use for foliar applications. And probably because silica is a mostly insoluble element, insoluble in nature, and the mobility in plants also very low. This is where we required a stabilized soluble chemical. That there we have to work on that area. That area of research is still in a government stage. Probably in future we may come up with a good formulations working together. Probably I think extensively it is used in Japan and China for several crops. And some of the crops like silica accumulating crops like rice and sugarcane, they are dominant and they're being mainly utilized. And the sources of residues of these silica accumulating crops also can be used as a source of silica for other crop nutrition. This has been applicable. It's already one product is developed in US Bangalore as a source of silica by using the rice husk. This is one area. And the importance of this silica, you can see, when you apply for a soil, probably the soil based through roots only it is absorption. So otherwise, either through leaves, shoots, or other products. And depending on the different absorption, different parts of the plants, you have different resistance capacity. You see, once the deposition of silica takes place during hull, probably that is a rice hull. So that probably it get transported and transported will be act as a very good water stress material. This is what happened. Certain diseases like rots and some fungal and bacterial diseases play a significant role in silica. That is one area where the silica deposition in terms of leaves it plays a significant role in detoxifying, maintaining the balance in the nutrition in the plant system. And probably the heavy metal toxicity which is contains or has been eliminated by the heavy metal toxicity by using silica. This is one area where we can really a positive nature. And another area where we can deposition in stem, where increases the strength and the rigidity and strength in the leaf system. That is one sheet. It will be helpful in the plant system. And especially in certain crops like rice, it prevents the lodging, and in case of even the low temperature, right, some of the hepatic stress and some of the and especially the phosphorus nutrition. Silica plays a significant role. Probably the silicates complexation plays a significant role in dissolution of phosphorus. This is several papers is available. In that particular area, it's one of the students can be taken up that work. That is also one area where you can pick up. Another is other elemental toxicity can be reduced in the in plant system as well as soil also. That is one area where we can. Then other most important is the composition between the aluminium and silica complex, especially very low soil pH conditions, so where we can detoxify aluminium content in the soil. These are some of the very important functions of this silica. And when we come to the plant system where silica, where accumulations takes place in the silica, this is the most important. See, we can see some of the papers are from China, just some of the references are taken from China only. China and Japan, they are the pioneers in silica nutrition, this is where it happened. Suppose when a applied silica to the soil, what happens when it is possible, it should be a soluble in nature, it's available only in the monospecific form. So it absorbs, it transports by diffusion as a nutrients or maybe by the mass flow. This is one area, two processes takes place in that. When the silica absorbs into the root system, what happens? It gets concentrated into the shoot system via a xylem. In such cases, what happens? A particular soluble monomeric form of silica, it gets polymerizes and it forms a silica gel type or silica salt type will be there. That salt or silica gel type, what happens? It forms a phytoliths. This phytoliths is the main thing which enters into the plant system. That plays a significant role in the plant system. This is where the main mechanism of silica acts on the host biotic and hepatic stresses. What we estimated is almost silica in phytoliths and cellulose and epithelial is almost 50 to 79% silica is there. But this circulation probably we don't achieve this much accumulation because because of low mobility, even if it is 50% accumulation in the leaf epidermal tissue, we can control, we can improve we can use the resistance against most of the pests and diseases. This is where we can achieve. Another most important aspect is that this silica mobility is agent by some of the promoters, or what we call it as transporters. And initially it was first identified by YSLIC, one of the G, and subsequently further one of the studies we have noticed that further uptake was silica was noticed by LSI second G. So now it is declared that both LSI1 and LSI2 express both in exodermis and endodermal fruits of different positions in the plant system. So we consider these are some of the transporters where the mobility of this plant is in the plant system. Another most important, we know the basic roles of the silica plays a significant role in two ways. It acts as a chemical barrier. The other one is a physical barrier. So mechanical barrier here means once the silica absorbs into the plant system or the shoot system or the leaf system, it forms a typical double layer silica gel-like layer immediately between the peticles, the leaves. What happens? This forms a rigidity, a strength it creates. Probably whatever the external pest, either it is a sucking pest or any other pest or aphids or this thing. Usually the certain pests like usually cheating and sucking pests, they will usually have single mandibles or they are variable mandibles. When it comes to the crop, it will only give us crack, scratches will give. So in such cases, if the ordinary enter into the leaf tissues, whatever the mandible will break. So this is one way mechanical barrier takes place. This is the one mechanism where you can control specific to the threats and something cheating and sucking pests. This is one area. And another thing is flower feeder also places will be controlled. Sometimes usually the xylem, we see xylem momentum through the xylem. But some case what happens when the flowing feeder also takes place. In such cases, some of the pests which are only target only the outer layers of the leaves. In such cases, that particular pest can be controlled by using silica. Then another most important is the defense mechanism. We kill it as a chemical method. Once the crop, when the silica accumulates in the system, either the shoot system or the leaf system or the root system, probably it plays a significant role in developing some of the organochemical methods or compounds. Some of the then which enhance, increase the phenolic compounds in the system. It gives a rigidity to the system. So in such cases, many fungal pathogens or any bacterial pathogens which hinders the attack on this because of rigidity strengthens the component. This is another mechanism which specifically is what I mentioned here. Some of the various enzymes like polyoxides, polyaluminum ammonia lysis and peroxidase. These are some of the organochemicals which plays a significant role which induce the resistance of the pest incidence. And another most important aspect, especially if you take up airway distress are different nature. One is salinity problem, water stress will be there, component. Then another one is high acidity or low acidity. This is one area. And these are the various airway distresses which also plays a significant role by silica influence. So here we can see that silica applications is also we have noticed that enhance stomatal conductance and carbonic sensation rates, and which plays significant ultra low structural organizations in the chloroplast. So suppose if it is accumulated with regard to the chloroplast, where the biomass under salt stress which shows an increment in biomass as well. Especially biomass production, especially in vegetable studies we have done it. Biomass production under salt stress we have noticed enormously high. And this probably because of structural inductions, and probably plays a significant role where water uptake with regard to the is being influenced by only the membrane restructuring. The role here takes place where water stress or any other hepatic stress is through the membrane restructuring by silica. This is one of the basic component that takes place. Then another most important beneficial effect of silica is also stress, which is increasing the root growth. Also, we have seen. And also leaf area index, food biomass, promoting land biomass, canicle and weight and honoring the number of tillers and yields. We have seen the some of the references we have seen in case of rice we see. And most other important is it regulates the leaf water loss. So what happened because of what I call blocking or thickening of the thickle layer, what happened the transpiration rate is drastically reduced. In such cases, what happens? The loss of water will be reduced. So probably the requirement of the withstand with drought conditions is more prone. So there again, silica plays a significant role. The thickening of the critical layer is most important. In order to achieve this, the mobility of the silica has to be essential. So to appropriate formulation is most important to mobility of the silica. This is where we required. Then another most important is it plays significant role in upregulation and downregulation. This is one area where it will modify the signaling pathways and antioxidant systems. Because of silica's role in membrane constitution, is that there are different mechanisms it happens. So these are details of the papers we have working on this signaling pathways and antioxidant systems also there. And these are some of the basic systems showing here for a plant defense response under drought conditions. See, based on this concept, what we have done in our IHR, because what we need a soluble nature and we require a stabilized compound. So we have taken industrial byproducts like potassium silicate as well as orthosilic acid, and we treated different aspects, different combinations with Arduino balls, and we combined with the different micronutrients like especially zinc and boron. And we developed a stabilized compound like both potassium silicate and orthosilic acid. And but at the same time, when you go for a foliar application, there should be we should maintain a pH of 5.5 to 6.5. Stabilizing this pH is most important. So there we have used some of the weak acids. Probably acetic acid is highly immature, unstable in nature. So now probably we are avoiding. Now we are taking up citric acid and salicylic acid, where we can stabilize the compound. Because we need a solution which should be suitable to the plant leaf. Otherwise, there will be a lot of scorching. Even a small rise in the pH or small low in the pH, crop will have a scorching effect. We have experienced this. So that's why we developed, we studied the stabilization studies with different compositions of sugar alcohol with these weak acids, and we put stabilizing agent and pH studies and admixtures, and we have developed some of the formulations, some of the usable nature of silicon compounds. Then we have also kept this for separation for 6 months. Then quarterly, with the low concentration, we started from probably 1 ml per liter, 2 ml, like this we have started working on that. Now we have stabilized the levels of the spray. Now we have stabilized up to 4 ml, we can use this solution and we can take up the experiment or we can have a further studies. This is what we have done last 4 years onwards. And we have taken up different crops and that is green chilies, capsicum we have studied, and tomato as well as onion also we are doing it. And with regard to the capsicum and chilies, we got a very good positive results. We got excellent biomass production irrespective of the sources of both either potassium silicate as well as orthospheric acid stabilized reagents. And we compared almost when we got to the with respect to the other elements what we use, we compared with the potassium chloride also, where that is also very significant with regard to the what we call disease residence incidence and other things. So we compared that as a what we call stable control. Then we have studied both orthospheric acid and we almost 18 to 22% yield increment of biomass production we have noticed both in chili as well as in capsicum. And marketable yield almost we got it excellent yield. Probably this is all because of what we call influence of silica and combination with zinc and boron, which is one influential effect. And we have seen the above also, even after this case, we got a good, especially we got the silica. Chili, we got highest fresh biomass we got it compared to other sources. While in capsicum, potassium silicate, we got relatively higher biomass. And accumulations, we see accumulations in the leaf compared to other parts of the plant is in the leaves only we got relatively higher accumulations in silica, up to almost 2% to 2.2% we got higher accumulations in the leaf. And probably, and also it is a significant role in enhancing the potassium, calcium, and calcium magnesium ratio. This is most important. And other aspects like uptake also we noticed, especially we got a very good uptake with response to the calcium, potassium, and also zinc. And whereas anyway, our what we call we have a root system below ground, probably we are not much accumulations we have noticed, probably because of probably low mobility in the root system. When we go for a foliar application, probably these might have taken more values. And with regard to these, what I mentioned here, this is what here this is specific, very rampant insects pests. This is thrips, one area where chili is very badly affected by this species. And we have drastically controlled pests by silica spray, especially orthospheric acid has a significant role in controlling the this particular thrips species. And the incidence was drastically reduced compared to the control. And with regard to the thrips, see mortality almost 50% reduction we have thrips. And but subsequently over a period of cropping period, a drastic reduction in thrips. Initially we have achieved 50% reduction. And subsequently the reduction is almost 25% to 80% we have noticed. So this is one very important observations we have noticed. Probably subsequently we have taken up even the capsicum. But another problem is another pest in onion is most important. That it is also we are working on that insects. Probably that is apex and white place. If we work on that, that's a problem. Probably the vector pest, most of the pests, most of the diseases which influence the vector pest means it's difficult to control. But when we control this vector, because this vector influences the leaf curl virus of the chili. So when we control this vector, probably we can control equally leaf curl diseases in the chili. And this is like also simultaneously we recorded different sources and different stages of the crop. And we found the same consistently these sources are drastically reduced thrips populations in the chickpea. This is in capsicum. And we come to the leaf curl disease also we have noticed, especially see when treatment with the orthosilicic acid is drastically reduced the leaf curl disease. Probably this is the control of this is vector leaf curl vector, and so this is one area where we can just concentrate. And anthracnose disease also we have drastically reduced where we control potassium chloride what we sprayed is relatively higher. We have noticed. Whereas the orthosilicic potassium silicate we have drastically reduced. And this is percent anthracnose. The chili is another problem where the crop, though the crop grows very well, probably anthracnose is another aspect where we can go for an hot pepper, hot chili concern. So, because of anthracnose, almost 30 to 40% yield loss will be there. In case if it is controlled, that is another 20% loss can be reduced. Probably still this is an initial study. Probably further, probably we will work on this anthracnose disease. Suppose if it works, this is one area where we can recommend for it may go on practice chili. Of course, the odor video is usually not much in chili, but however we have noticed odor video and virus infections are relatively less. Even odor video in capsicum also we have shown it is comparatively less. Even silica probably and further we worked on this how this is effects of that probably. But we know the membrane alteration, membrane resistance is when where weight is stable. But due to accumulation of silica, hence the production and detoxification of oxygen radicals and hydrogen peroxide. This is where we have studied. So NDA and hydrogen peroxide values are drastically come down, whereas other enzyme systems like SODs, catalysis, and other peroxidase enzymes are showing relatively high. In such cases, what happened? The phenolic compounds in the leaf membrane system is considerably higher. So further, it will intensify the lignification processes. If lignification process increases, the synthesis of lignin more. When the lignin synthesis is more in the leaf system, probably the incidence of pests probably either this may actually induce resistance to most of the pathogens. What either is fungal disease, probably in Sagataga, probably in banana they have observed the induced resistance by silica it is observed, especially the Sagataga disease. Whereas here we have noticed some of the is probably another most important is phytoalexins. This is some of the formations of phenols is most important. This has to be studied in detail. But say onion and tomato is another audience because silica work has been worked initially in India as early as 1943 itself on tomato, but not much progress has been done by silica because silica has been considered 1940s only as an beneficial element. It is not recognized much in the world. Probably now, because of advance in science, probably the countries like Japan and probably in China, the usage of most of the agrochemicals they are drastically reduced. So the alternative source of combating these pathogens they have established and they have come out. So that's why probably the science has developed, especially Japan. They are really they have forward, I mean, they have worked on silica extension techniques. We have also studied that when silica sprayed, we have taken a cross-sectional and bisectional leaves we have taken. How? Because the mechanical barrier is also another area where we have to study. Because whether when we spray silica continuously, whether this silica accumulation takes place immediately beneath the critical layer, that is most important. Otherwise, just we are spraying will not do. So, in order to study that, we have taken same. So, we have taken a both cross section and vertical sections of leaves. where we studied and we have identified some of the epidermal cells, and we have identified by using cell there are two types of bodies we have noticed. Silica in the leaf tissues, either it will be we have seen spread isolated, or it will be accumulated as a ladder-like structure. This is what this scattered ladder-like structure we have noticed. Here we can see here. This is where a ladder-like structure in the first treated one, and whereas the previous one, this silica is spread isolated in the leaf system. Especially these silica is accumulated only in certain specific cells, epidermal cells, like rod cells and some of the silica monofilament cells and bulliform cells. There are two types in adaxonal epidermis as well as there are different levels of epidermal cells are there. In such cells, we have noticed these silica compounds, and there, and also again we have studied by using this studies we have studied the structural rigidity. Because once the silica accumulates between the epidermal cells, rigidity of the strengthening of the leaf texture is most important. What is the normal strength of the leaves and the silica sprayed leaves also we have studied. You have noticed some of the structural rigidity are 17 times higher than the leaf we have noticed. This is another very good information what we got, and this paper is accepted in American Chemical Society is having an impact of 15.83. This is a well recognized study in our study we have recognized. International impact is 11.3, which is one of the findings it is done. So that rigidity that induces resistance. So this is what we have done this work with regard to different data. Finally, when you say we conclude actually, silica peptides conjugate silica salicylic acid throughout the plant body. This we have noticed both the roots, stem, shoots, and leaves. And silica fertilization by industrial silicates, a low-cost industrial byproduct, has a significant role and very important role in these mitigating these stresses. And probably in future, probably we may have an alternative source of these fertilizers. Because today, our sources, or we have a very limited resources, especially with regard to the phosphorus and potassium fertilizers, and even nitrogen also. Nitrogen probably unless we tap the ammonia from the environment, probably most of the fertilizer industries, most of the fertilizer plants are very good. They are not investing in the new fertilizer factory. Because government already decided importing fertilizers from other countries is cheaper than manufacturing fertilizers in India. In addition, we don't have any resources. Phosphorus sources are not there. We have exhausted. Potassium sources are there. Today, India 100% phosphatic fertilizers, potassium fertilizers we are importing from other countries. So if DAP, if the supply of DAP is not available in India, our agriculture also will be badly affected. Only most of the DAP is imported from other countries. This is what it happened. So silica also plays an important role as an ordinary source of phosphorus, and sodium can be used as an ordinary source of potassium. These areas already started working on that. That's why present government is promoting nano urea and nano DAP because of this. Because our Indian exchequer is going higher side for importing these fertilizers. And besides now in US and other European countries, silica is already been recognized as an organic crop production, as a source of organic crop production. And India, based on that, India has come out with gazette notification is an organic fertilizer, not the best organic source of fertilizers. And And now in America it's already using, usually green modes are quite common in the temperate countries. So now this potassium silicate is extensively used because it doesn't give any residues export, because that's why in India for zircon they are using silicon because because of the rigorous valid export conditions, this doesn't give any residues for the controlling of pesticide. That's why they are using, now it is that's why they are waiting for government of India to consider as an organization. They have declared an amount of extension using. This is very rapid. This is another other examples, other important sources of silica is consumption of silica is not harmful to the human being. Like zinc, among the nutrient elements, zinc and silica both are less for human consumption. Even excessive consumption of silica also is not harmful for us. And it is, moreover, it is required for our body, especially bones. See, we are blessed with because we have strength with bones because we are the rice eaters, and rice and the sugarcane are the silica accumulating crops. So we are the more consumption of silica. Probably that's why the daily we are meeting daily requirement of silica, almost 30 milligrams of silica we are required for an average human being. Whereas in European countries and other countries, they are not rice eaters, only bread eaters. So in cases only they supplement to beef. The requirement for per day requirement of a silica is the stabilizing agents, some of the that is poly sugar or first we can use, and weak acids. So we can add solubility, but after soluble, what happens, it will get accumulated, coagulated. It will get coagulated. Addition of any other element will get coagulated. So in such coagulated, what happens? There will be a solid portion left over in the lower portion after keeping for some 1 month or 2 months. So you can decant the solid portion. You can use the supernatant liquid. That is, then you realize the silica content in that supernatant liquid, and based on that you can monitor. Dosage liquid test based on the silica present in the supernatant liquid. This is only one thing, but in the market commercially available silica sources are available. What they do, they mix with humic acid, then potassium humate, then amino acids, seaweed extract. All this, even the silica also acts as a biostimulant. So all these biostimulants, we cannot quantify which part of the chemical is contributing which what. This is what commercially available, it's already available formulations. But that's what we are trying to come out with only silica portion, that is most important. If we come out, what is the role of silica exactly to understand, probably in future we can develop some formulation combination that is amino acids, seaweed. And usually given humic acid also they are used.
So in your formulation, you hold some weak acids.
Yes.
So that you are using for stabilization or
pH adjustment. Because one year application, we cannot spray less than 5.5, and we cannot pH adjustment go more than 6.2 or 6.5 because it will not be effective. If it is less, means it will be costly. If it is more, also probably it will not be effective. You know, to increase the efficiency of the solution, we are using.
So among the weak acids, you say citric acid is.
Citric acid is best source. Best source we can manage this. We can suppose it probably the pH will be in acidic side of it. Relatively higher than 7 pH or still there. Then we will go to pH. Then stabilization is required. For stabilization, using stabilizing agents, sodium metaoxide, some of the stabilizing agents are there. Those is can be some of the poly sugar alcohols also. This is used for your micronutrient formation, so we can use.